Hi guys, welcome to Team's channel. This is the fifth biochemistry lecture. In the previous 4 lectures we have talked about the glucose metabolism. In this lecture we are going to start the glycogen metabolism, which is a polymer of the glucose. Please remember that glucose is a very important energy source for exercising muscles, and we can obtain the glucose from diet, glycogen degradation, and gluconeogenesis. However, during fasting, when we cannot obtain glucose from diet, the liver glycogen stores are degraded, so that glucose is rapidly released into the blood in order to maintain blood glucose level. This is known as homeostasis. Also, the muscle glycogen is degraded. However, it is used to provide energy for those exercising muscles, and this means the muscle glycogen is not involved in the homeostasis. What if the glycogen stores are depleted? Now our cells are going to synthesize the glucose from gluconeogenesis. Here are important notes that you need to know. The liver glycogen is very important for maintaining the blood glucose concentration during fasting, especially from 16 to 18 hours of fasting. And please remember, 10% of the total liver weight is made of glycogen. However, this liver glycogen stores increases during well-fed state. Well-fed state, our blood glucose level increases. So, here the liver acts as a storage organ, where it uptakes the extra glucose from the blood in order to be stored in the form of glycogen. That's why glycogen storage increases during well-fed state. During fasting, it decreases. Why? Because glycogen would be degraded to provide glucose that would diffuse out from the hepatocytes to the blood to maintain the homeostasis. Muscle glycogen storage is very important to serve as energy for ATP, or as a source of ATP during muscle contraction. During muscle contraction, the glycogen would be degraded into glucose. The glucose would enter either aerobic glycolysis or anaerobic glycolysis in order to produce ATP that would be used by the myosin heads to be hydrolyzed, providing energy. This energy is required by the myosin filaments to pull the actin filaments to initiate muscle contraction. 1 to 2% of the total muscle weight is made of glycogen. However, remember that our body is mainly made of muscles, so the total weight of skeletal muscles in our body is more than that of the liver, so that most of the glycogen is found in skeletal muscles. This storage is not affected by short periods of fasting. Why? During short periods of fasting, the muscles are not involved in homeostasis. However, in prolonged fasting, they decreases. Why? Muscle glycogen storage are not involved in the homeostasis, but here the glycogen is degraded to provide ATP for the muscle contraction. However, we cannot regenerate the glycogen because of the prolonged fasting. That's why it decreases. What is the structure of glycogen? It's a branched chain polysaccharide which is made of alpha-D glucose. Alpha-D glucose molecules inside the chain are connected together using alpha-1,4 linkage or alpha-1,4 glycosidic bond. As we mentioned, it's branched, so these branches would connect to the main chain using alpha-1,6 linkage. Where can I find the glycogen? It's found inside the cytosol of the cell as large spherical cytoplasmic granules that contain enzymes for glycogen synthesis. This can be considered as adaptation because this will provide the enzymes whenever they are required. Now let's start the synthesis of glycogen, or to be known as glycogenesis. In this process, glycogen is synthesized from alpha-D-glucose using ATP and UTP. Actually, it occurs inside the cytosol. The first step, the free glucose inside the blood, will be uptaken by the cells. Now, the free glucose is going to be phosphorylated to glucose 6-phosphate by the help of hexokinase or glucokinase in order to trap the glucose inside the cells. Now, glucose 6-phosphate is going to be converted or isomerized to glucose 1-phosphate by the help of phosphoglucomutase enzyme. Now we are going to synthesize UDP glucose from glucose 1-phosphate and UTP by the help of UDP glucose pyrophosphorylase. The coming step would be elongation. We are going to add other glucose residues. This is catalyzed by glycogen synthase, which synthesizes the alpha-1,4 linkage inside the glycogen. Unfortunately, this enzyme cannot initiate a chain. It only elongates a pre-existing chain of glycogen because it needs a chain at which it binds and moves on to be able to synthesize the alpha-1,4 linkage. So the cell now requires to make a primer, which is a fragment of glycogen or a very short chain of glycogen, which is made up of at least 1 to 4 glycoside residues. We are going to overcome this problem by the help of glycogenane, which is a homodimeric protein. This protein is going to serve as acceptor of the glucose from UDP glucose, or we can call it an enzyme since it catalyzes the formation of the primer. As I said, it transferred 4 molecules of glucose from the UDP glucose to the non-reducing end of growing chain. Non-reducing means the terminal end or the terminal residue. Now I got a very short chain which is made up of at least 4 molecules of glucose. This short chain is going to serve as a primer. This primer is going to be bounded to the glycogen synthase and elongated by glycogen synthase. Now we need to branch the chain. Remember, amylose have a very similar structure to the glycogen. However, it is unbranched while glycogen is branched. The importance of branching is that the branching increases the solubility of any molecule. That's why the glycogen is more soluble than the amylose. The branching process is catalyzed by 2 branching enzymes, amylo alpha-1,4 and alpha-1,6 transglycosylase. These enzymes are going to remove 6 to 8 glycoside residues from the non-reducing end. And they are going to attach these residues to any non-terminal residue. For example, try to imagine this is a chain. We are going to remove from 6 to 8 residues, and then we are going to add it to non-terminal residue. Actually, any one of them. Non-terminal means not at the very end. So, the end result would be a branched polysaccharide known as glycogen. The opposite occurs during fasting. Degradation of glycogen or glycogenolysis. Here, the stored glycogen of the liver cells or skeletal muscle cells is going to be degraded. The degradation occurs by the breaking of alpha-1,4 glycoside bonds by glycogen phosphorylase, leading to the production of glucose 1-phosphate. This process is going to be continued until 4 glycoside units are remained on each chain of the molecule, and the remaining product is known as limit dextran. Now I have only 1,4 residues found in either side of the chain. Here is a chain, and here are 4 molecules, and on the other side 4 molecules. Now I'm going to remove the branch by enzyme that have 2 active sites. That's why we call it bifunctional enzyme. The first active site of the enzyme, known as alpha-1,4 leukotransferase. This active site is going to catalyze the removal of 3 residues from one branch and is going to transfer these residues to the other branch, so that this branch is made of 7 residues, while this branch is made of only 1 residue. The 7 residues on the other side are going to be degraded by glycogen phosphorylase until again only 4 residues are maintained. Why? Because glycogen phosphorylase only works on long chain. However, remember that only 1 residue was remained on this side. This residue is going to be removed by the breakdown of alpha-1,6 linkage by the help of amylo alpha-1,6 glucosidase activity. This leads to the production of 1 free glucose molecule. This cycle is repeated until the process is terminated. Now I got glucose 1-phosphate and free glucose. Free glucose is going to diffuse out of the cells, especially liver cells. This occurs in the liver cells. In order to maintain homeostasis, while the glucose 1-phosphate is going to be isomerized to glucose 6-phosphate by phosphoglucomutase. This glucose 6-phosphate is needed to be dephosphorylated, so that is going to be transported into the endoplasmic reticulum of the liver cells by glucose 6-phosphate translocase. Inside the endoplasmic reticulum is going to be dephosphorylated to give me free glucose. This is catalyzed by glucose 6-phosphatase. Then, of course, the glucose is going to leave the endoplasmic reticulum and enter the cytosol. Inside the cytosol, the glycogen or the glucose actually is going to leave the hepatocytes and get into the blood to maintain blood glucose level. However, inside the muscle cells, the glucose 6-phosphate cannot be dephosphorylated. Instead, it would enter either aerobic or anaerobic glycolysis to provide me with energy needed for the muscle contraction. And this is the way of degradation of glycogen. However, there is another method of glycogen degradation which is known as lysosomal degradation. A very small amount of the glycogen, which is 1 to 3%, is degraded by lysosomal enzymes and acid maltase. And any deficiency in these enzymes would lead to glycogen storage disease number 2, to give me a disease known as pump disease. This means after the trappement of glycogen inside these lysosomes, deficient enzymes mean that the glycogen will not be degraded. So this glycogen will be accumulated inside the vacuoles of the lysosomes. In order to recap, in this lecture we had the synthesis of glycogen, some important notes for glycogen, degradation of glycogen, and glycogen storage disease number 2, which is known as pump disease. And here we have reached the end of our lecture, and good luck.